Hi guys, it's Francis here, and I'd like to continue our discussion of proteins. Proteins are 3D structures. They are conformationally different from one another, and it's this conformational difference, this difference in shape, that determines each protein's function. So let's look at some different proteins that have unique functions. For example, antibodies are part of our immune system. The antibody has a particular shape that's going to recognize a particular foreign substance, a specific pathogen, to get it out of our body. Enzymes, remember lock and key theory way back when you took living environment? Enzymes act as a lock and a key. They bind to specific substrates where if I don't have the right key, I'm not going to unlock that chemical reaction. Neurotransmitters pass signals from one to another by binding to receptor sites that have a specific shape. So if one neurotransmitter can't fit into the correct receptor, that signal doesn't get sent. In this video, Stephanie Castle summarizes the most important protein functions. There are 4 structures to a protein. The primary structure, which is sometimes abbreviated like so, the primary structure just refers to the unique sequence of the amino acids. Which comes first, which comes second, and we know that methionine is always going to come first, but after that, the possibilities of different arrangements is really almost infinite. If I change even one of those amino acids, in other words, I remove one, or I substitute one, or I add one, it can change the conformation, it can change the shape of that protein, therefore altering its ability to function. For example, sickle cell disease occurs when one amino acid is substituted for another one, and it results in this awful disease that affects the hemoglobin of someone. So it affects the ability of that person to carry oxygen throughout their body. These people don't live very long. They often die between 30 and 40, and they are tired all the time. They don't have the oxygen needed to do their daily functions. So as you can see, the way someone might get sickle cell is just one substitution where valine is in the place of glutamine acid. Here's a video, Stephanie Castle talking about the importance of the sequence of amino acids in polypeptides and what effect that would have on the polypeptide. So once our amino acid sequence is determined, we move on to our secondary structures of a protein. And this is when the shape starts to take hold, where I'm going to coil into either alpha helices, alpha helix, or I'm going to fold into beta pleated sheets, where they are held together, that alpha helix is held together, and then the beta pleated sheet is held together by hydrogen bonds at regular intervals along that polypeptide backbone. For example, silk is a protein that is extremely, extremely strong. In fact, some researchers have been looking into implementing spider silk in bulletproof vests for the military. The next structure is the tertiary structure, and this is when our R groups start interacting. Remember, our R groups are varied between each one of our amino acids, so there are going to be different types of interactions within each polypeptide. So you might have polar R groups interacting with each other via hydrogen bonds, or charged R groups interacting via ionic bonds. You can have hydrophobic interactions and van der Waals interactions. Furthermore, you're going to have these disulfide bridges, where you have a covalent bond, which is strong, forming between the sulfhydryl groups of cysteine monomers. So this is what stabilizes this structure. Lastly, when I have R group interactions between different polypeptides, I start to form my protein. There are different types of proteins. You have fibrous proteins and globular proteins. Collagen is an example of a fibrous protein. It kind of looks like a braided structure, whereas a globular protein, I kind of think glob sounds like the word blob. Well, they're kind of blob looking. Hemoglobin is a classic example of a globular protein. Hemoglobin is also unique because it's considered a conjugated protein. A conjugated protein consists of a protein with a non-protein substance. So in the case of hemoglobin, it's got iron that is attached to the protein. So here, you've got your primary structure. Your primary structure is just the amino acid sequence. When we move on to our secondary structure, you have your alpha helix and your beta pleated sheet. Now our polypeptide is starting to take shape. The tertiary structure is when your R groups start to interact, and they're going to have hydrophilic interactions, van der Waals interactions. You'll have hydrophobic interactions. You'll have ionic bonds between charged R groups. Then we get to our quaternary structure, where you have R group interactions between polypeptides. So one polypeptide interacts, or one polypeptide's R groups interact with the next polypeptide's R groups. Stephanie Castle does a really good job of summarizing protein structures. So here are the answers to the protein structure review questions, where secondary structure gives us those alpha helix and our beta pleated sheets that are held together by hydrogen bonds. The sequence of amino acids is our primary structure. 2 or more polypeptide subunits, that's when you get your quaternary structure, your final protein. Hydrogen bonds, ionic bonds, hydrophobic interactions, van der Waals forces, disulfide bonds, those are all going to be found in your tertiary structures and your quaternary structures. These are the interactions between your R groups. And your secondary structure includes alpha helixes and beta pleated sheets. Next time that you'll see me, we'll be discussing enzymes, which are another important type of protein. I hope this helps.